This is Meiosis Part B. In the previous meiosis video, we looked at what happened when one chromosome went through meiosis. Today, we're going to do something a little bit more complicated and look at one cell, one nucleus, that has two different chromosomes in it. So we're going to look at a diploid organism. So two copies of every chromosome, the pink and the green. And we're going to continue with the same chromosome numbering. Chromosome 1 from dad, the paternal allele or version of that chromosome, and the maternal version of chromosome 1. And then we have the paternal and maternal versions of chromosome 2. The first step that we need to consider is the synthesis of new strands, new double helices of DNA. And that makes us our tetrads. 4 copies of each chromosome. 2 chromosome 1s from dad, 2 from mom, and the same for chromosome 2. Now at this point we have the 2 tetrads lined up in one of the first cell divisions that's going to take place. The first of 2 cell divisions is going to happen, as usual, down the middle of this cell. An important point to make about meiosis with 2 or more chromosomes is that when we're looking at 2 chromosomes, there's an alternative arrangement of the tetrads across the cell. So up here, if I continue the same color scheme, these are not extra DNA molecules, I'm just replacing with the colors that I'm using for tracking the different chromosomes, pink for chromosome 1 and green for chromosome 2. You'll notice we have the paternal versions of the chromosomes on the left side and both of the maternal copies of each of these chromosomes on the right. So there's another arrangement of these chromosomes that can occur. So we still have the tetrads, all of the chromosome 1s lined up next to each other and all the chromosome 2s lined up next to each other. But in this case, we might have the paternal versions of chromosome 1 there and the maternal on the other side. The reverse, though, to the first example might happen here on the bottom, where we'd have the maternal alleles on the left side of the cell and the paternal on the right. And during the first cell division, I'm going to say that that cell is going to divide right there. And this difference, whether or not the 2 paternal versions are on the same side of the cell, or it's paternal with maternal and paternal with maternal, is going to make a big difference in the types of gametes that are produced at the end of meiosis. So after the first cell division, in each of these, this will generate not necessarily oblong cells, but we'll have the chromosome 1s and chromosome 2s arranged thus. So we have the same genotypes in both of these cells. We have the paternal versions of chromosome 1 and the maternal, paternal, maternal. And the opposite again here. I'm just being explicit about what these cells and their haplotypes and genotypes look like after the first division of meiosis. Now, here's where the real difference occurs in the second division. So each of these 2 cells in the top and each of these 2 cells in the bottom will divide a second time to produce the 4 haploid gametes. In this case, divisions occur again in between, down the middle of the cells, but this time in between the sister chromatids. And this dictates the types of chromosomes that we get in the gametes. So each gamete will receive one copy of each of the two chromosomes, but the haplotypes, the genotypes of the haploid gametes, are going to differ depending on how these tetrads were arranged before meiosis I. So you can follow the alleles through after the division and discover that here we have the paternal versions. That would be products of this cell division, and the two maternal haplotypes together. So those are the two cells that result from this cell dividing. And down here now we have the paternal with the maternal. That's products of that cell division. And we have the maternal chromosome 2 with the paternal chromosome 1, and that's products of that cell division. The really critical take-home point from all of this is that we have an equal number of alleles. We started with 1 to 1 to 1 to 1. 25% of these chromosomes were one paternal, one maternal, and so forth. We still have that same ratio here. We could count the number of green paternal alleles, 1, 2. We could count the number of pink paternal alleles, 1, 2. And then we could do the same thing for the alleles that we see down here in this other round of meiosis. And we see that we have more green paternal, so that takes us from 2 to 4. And the same is true of the pink paternal. And then when we tabulate all of the maternal copies of these chromosomes, you see that in these 8 myocytes, either sperm or eggs, there are also 4 copies of each of those. So we have a total of 16 double-stranded chromosomes, molecules of DNA, and they're in a precisely 1 to 1 to 1 to 1 ratio. 25% of all of the chromosomes here are each of those chromosomes. What's incredibly important is that those frequencies are maintained in the combinations of which chromosomes exist. So for example, we have a quarter, or 25% of our gametes have the green paternal and the pink paternal chromosomes 1 and 2, together in the same gamete. So that gamete's haplotype is different than this one. 25% of those gametes have the maternal versions of the pink and the green chromosomes, and here a quarter of the gametes have the maternal green but the paternal pink chromosome, and the reciprocal is true in this bottom set. This is the essence of Mendel's second law. Mendel's second law is the law of independent assortment. And what this means is this is the independent assortment of haplotypes into gametes after meiosis. Put another way, this tells us that 50% of all of these cells here have the green paternal 1, 2, 3, 4. Half of the 8 gametes have the green paternal version. But the independence comes in what happens to the other chromosome, the pink chromosome. Of those 4 different gametes, half of those 4 have the pink maternal version of the other chromosome. The other half have the pink paternal allele. So that's independence. A quarter of all of the gametes have a particular allele of chromosome 1, and then which allele of the other chromosome they inherit is entirely independent. So you get copies that are PP, paternal-paternal, and paternal-maternal. Now, the question I'd like to leave you with to answer for our next class is, what would the meiotic products be from an individual that has, say on gene A, allele 1, this is a diploid individual, and they're heterozygous, so their other chromosome has the allele number 2 at the A gene. And this individual also has a B gene on a separate chromosome, the semicolon, and they're homozygous for those alleles. So again, what would the meiotic products be after meiosis of a cell with that genotype? And we'll discuss that in our next class.